friends, today let us see about packaging in DNA helix. This is for class 12, chapter 6, molecular basis of inheritance, under the packaging of DNA helix. For 12th standard, chapter 6, molecular basis of inheritance, under the packaging of DNA helix. So in prokaryotes, for example, E. coli, Escherichia coli bacteria in prokaryotes, see, they do not have well-defined nucleus. Actually, prokaryotes do not have well-defined nucleus. Prokaryotes, they are primitive nucleus, primitively formed nucleus. Okay, prokaryotes. Karyo refers to nucleus. Pro means very primitive. They do not have nuclear membrane, nucleolus. You must have studied in your lower class. So, no nuclear membrane, no nucleolus. Hence, it is called nucleoid. It is called nucleoid. They are called nucleoid. They do not have well-defined nucleus. Okay, prokaryotes do not have well-defined nucleus. DNA is not scattered. DNA is negatively charged. It is held with some proteins, okay, that have positive charges. DNA is not scattered. DNA is held with some proteins that have positive charges. The DNA in nucleoid, that is in primitive nucleus, is organized in large loops held by proteins. Okay, they are looking like large loops and they are held by proteins. You can see a picture here. It's a large loop. Okay, these dots refer to positively charged proteins. Positive charged proteins, large loop. Okay, and this is the negatively charged DNA. This is how the DNA is packaged in case of nucleoid in the prokaryotes. Okay. So let us see the packaging of DNA helix in eukaryotes. Packaging of DNA helix in eukaryotes. In eukaryotes, here the organization is complex, more complex. The positively charged proteins are called histones. Positively charged proteins are called histones. Histones are positively charged. These proteins, that is histones, they get their charges, acquired charges from amino acid residues like lysine, arginine. So these lysines and arginines are positively charged. So proteins, that is histones, get charged from amino acid residues. For example, from lysine and arginine, which are positively charged. Histones are organized to form 8 molecule structure called octamer. They are organized to form 8 molecular structure called octamer. Packaging of DNA helix in eukaryotes. The negatively charged DNA. Okay, the negatively charged DNA is wrapped around the positively charged histone. Okay, it is wrapped around the negatively charged. The negatively charged DNA is wrapped around the positively charged histone. Histone octamer. Octamer is because it is of 8 molecular structure. They form a structure called nucleosome. The negatively charged DNA is negatively charged. It is wrapped. Okay, it surrounds around the positively charged histone octamer. Histone are basically proteins. They are positively charged. Okay, this structure is called nucleosome. Here you can see the picture of nucleosome. This round structure is the 8 molecule histone octamer. You can see DNA is surrounding this octamer. Okay, so DNA is always negatively charged. Protein is positively charged. This 8 molecular structure is called histone. That positive charges come from amino acid residues. Okay. Normally, a typical nucleosome contains 200 BP. BP means base pairs, that is adenine, thymine, guanine, cytosine. That kind of pairs are called base pairs, ATGC combination. Okay. So refer the DNA molecular structure: adenine, with double bonded thymine. Similarly, guanine with cytosine, triple bond. The structures, remember, that is called base pair. 200 base pairs of nucleus you can see in a wrap. Then chromatin or thread-like structures. Again, refer your primitive or nucleus diagram. When you draw nucleus, you'll have a thread-like structure called chromatin. Okay. So the chromatin or thread-like structures, they'll be seen as beads on string. Okay, like small beads on a string under electron microscope. It gives an appearance like beads on string when you observe under electron microscope. That is how in nucleus it will look like. But at the time of cell division, these chromatin thread-like structures, these get coiled, condensed, and become chromosomes and get ready for cell division. They become coiled, condensed, and form chromosomes at the time of cell division. So here you can see the picture of histone. This is called a nucleosome. Okay, that DNA that wraps has 200 base pairs. That is adenine, thymine, guanine, cytosine combination. 200 base pairs. Okay, how the chromatin will look like beads on string. Okay, for chromatin picture, see the nucleus diagram. That the thread-like structure on the nucleoplasm. That is called chromatin. We call it as a reticulum, chromatin reticulum, because it is a network of thread-like structures. So they appear as beads on string when observed under electron microscope. EM means electron microscope, right? And at the time of cell division, they become chromosome. They become coiled, they become condensed, short, and form chromosome-like structures. So let's see about non-histone chromosomal proteins, NHC, non-histone, okay, chromosomal protein. So here are two types, euchromatin, heterochromatin. Euchromatin, heterochromatin. Just three differences. Euchromatin are loosely packed. They are packed very loosely. Heterochromatin are densely, very thickly packed. Euchromatin stains light. Okay, they do not accept the stain that much. It stains light. Heterochromatin, it stains dark. They accept more stain. Achromatin transcriptionally active, very active, and heterochromatin, they are inactive. Okay, so that's about the difference between euchromatin and heterochromatin. So there we come to the end of this particular packaging of DNA helix. In my next video, let us see the next topic. Thank you.